Many tumor suppressor genes are present in our cell, but for NEAT, only two are extremely important. First one is p53 gene, popularly known as guardian of the genome. This p53 gene produces p53 protein, which actually involved in tumor suppression. Second gene is RB gene, also called as retinoblastoma gene. This RB gene produces RB protein, which is involved in tumor suppression. Let us start with most famous tumor suppressor gene p53, the guardian of genome. p53 gene is the most important tumor suppressor gene. It produces p53 protein, which monitors DNA damage and prevents abnormal cells from dividing. p53 protein performs four major roles in cell. First one is detecting DNA damage. In this, p53 constantly monitors the DNA. When DNA damage occurs due to any carcinogen, this p53 gets activated. Then second function is stopping cell cycle. p53 stops cell cycle at G1 checkpoint. I repeat, p53 stops cell cycle at G1 checkpoint. This prevents damaged DNA from being copied and passed to daughter cells. Third function is activating DNA repair. p53 activates DNA repair enzyme. These enzymes fix damaged DNA. After DNA repair, cell cycle resumes normally. The fourth function is inducing apoptosis. Sometimes DNA damage is too severe and cannot be repaired. So, at that time, p53 triggers apoptosis, that is programmed cell death, which leads to death of all abnormal cells. Now, the question is, if p53 get mutated, then what will happen to cell? So, if p53 tumor suppressor gene is mutated, it loses its normal function. Firstly, this mutated p53 cannot detect DNA damage in the cell, and no detection means no repair of DNA damage. This will continue cell cycle, and damaged cell will synthesize constantly without cell cycle arrest. These mutated cells then multiply rapidly and accumulate more mutations, which ultimately leads to development of tumor, that is uncontrolled cell growth, that is cancer. Important point to be remembered here is normal p53 acts as a protector of cell, but mutated p53 cannot provide this protection, so it leads to cancer development. In this image, we can see that there are different types of cancers in which p53 is mutated: breast cancer, lung cancer, colon cancer, brain, liver, and leukemia. Let's quickly revise high-yield NEAT facts. p53 is called as guardian of the genome. It is tumor suppressor gene which arrests cell cycle at G1 checkpoint. It activates apoptosis in case of severe DNA damage, and we have found that around 50% of human cancers involve mutations of p53 gene. This is all about p53 tumor suppressor